Good evening. I hope you can hear me. Okay, so we'll start the class now. So again, this week we'll be looking at sample problems that are of relevance with the lectures of week 3. So we'll go to the first question. The question is, match the alkaloid on the left-hand side with the appropriate pharmacological action on the right-hand side. And the options that are given are, on the left-hand side, it's morphine and vinblastine. And on the right hand side, it's analgesic, stimulants, and antileucemic. So you can take a couple of minutes and then start by uh, describing what analgesics are and what antileucemics are. Then we'll look at the right answer. You can take a couple of minutes and then unmute. Yeah, you can unmute. Do you know what analgesics are?
Sir, I think analysis means it reduces the pain, sir.
Okay, and stimulants and antileucemics?
I don't know, sir.
Okay, so like you mentioned, analgesics are pain relieving compounds, whereas antileucemics are anti-cancer agents. And yeah, so do you know about vinblastine and morphine by any chance?
No, sir.
Okay, so morphine is it's an opioid. It is derived from poppy plant. So this is an alkaloid that has this analgesic property. So it binds to the receptors in the central nervous system. So one would not be able to perceive pain, and which is why it is used in hospitals to reduce the degree of pain. Whereas vinblastine, it's used as an anti-cancer agent because it can prevent microtubule from forming, and hence you know that microtubule is needed for division of cells. And in cancer, the existing problem is cells divide at a rapid rate. Because when blastin binds and prevents microtubule formation, it can be used for inhibiting microtubule formation and hence cancer cell division. So, among the options here, Vinblastine is an anti-leukemic, and morphine is an analgesic. The next question is, indole provides the fundamental skeleton for indole alkaloids. Identify the basic structure of indole from the following. And they've given four different structures. You can take a couple of minutes and then you can try to maybe mention each of the compounds here, what the structure is called as, and then we can go to indole alkaloids. You can take a couple of minutes. Okay, so here, if we look at this, there are four different kind of structures given. You might be familiar with option B. It has a benzene ring with an OH group. This is called phenol. This might also be familiar. This has two rings with an NH here, and it's present in nucleotides. So you can try thinking about this. Whereas option C and D are uh, something that you might not have studied earlier. So indoles in general are bicyclic, meaning it contains two rings. So here, uh, mm, this benzene ring is fused to a pyrrole ring, a five-member ring that contains a nitrogen, and this is the indole that we are looking for. Like we mentioned, option B is phenol, benzene with an OH group. Option C is a derivative of pyridine, which contains a six-member nitrogen-containing ring. And option D here, it's again bicyclic because you have one cyclic ring here and a heterocyclic ring here, just out of this plane. But that's not the skeleton that we are looking for. Right, we'll go to the next question. Tropane alkaloids found in Atropa and Hyoscyamus are biosynthesized in roots. Identify the specific tissues in the roots that are associated with the biosynthesis. And four different options are given here: cortex, endodermis and cortex, endodermis and pericycle, and vascular bundle. Okay, you can take a couple of minutes and start by mentioning what propane alkaloids are. We'll take it from there. We'll look at where exactly it is synthesized and what the components here play a role in.
Sir, I think it's option D, sir. Okay. So propanol alkaloids are bicyclic alkaloids. These are secondary metabolites. So, okay, sir.
Till hyoscyamine. Now, which gene needs to be transferred to the plant so that the target plant can produce scopolamine? So, for this, we need to know what hyoscyamine is, what scopolamine is, and what is the pathway. Four different options are given here: a hyoscyamine 6 beta hydroxylase, polyketide synthase, putrescine methyltransferase, and tropinone synthase. You can take a couple of minutes and maybe look at the pathway involved here so that you know how hyoscyamine and scopolamine are related. We will also look at the pathway and then figure out which of these enzymes is required. Yes, Swati, you can unmute and you can first tell us what you know about hyoscyamin and how it is related to scopolamine.
Yeah. Uh, so if the pathway is propionine will be synthesized to tropane and
Cyamin?
then hyoscyamine and then it will lead to the scopolamine production. So which gene need to be transferred? I think
Do
in
you know the
hyoscyamine,
structure of hyoscyamin and scopolamine? Chemically, what is the difference between hyoscyamin and scopolamine?
see, this one hyoscine is the atropine alkaloid, and scopolamine is again another name of the both are the anticholinergic. No, hyoscine
Yeah, yeah, yeah.
also,
So
yeah,
both of
scopolamine are both
these
anticholinergic. Yeah,
both of these have very similar properties. At the chemical level,
atropine, they're synthesized from atropine. Yeah,
yeah, at the chemical level, they'll have a very small but significant change, such as a methylation or hydroxylation. If you know that, you'll be able to tell us which of these enzymes will be involved. So,
I think that option A, hyoscyamin, the hydroxylase enzyme will be involved because this putrescine is another alkaloid which will be um, option A.
okay,
Is
you
it?
think, okay, we'll check with Shrestha as well. Shrestha, do you go with what Swati says, or do you have a different answer in your mind?
I don't know, sir.
Okay, no issues, no issues. We'll look at the pathway first. So, all of this starts with ornithine, the amino acid, and from ornithine we get putrescine. And this enzyme, putrescine methyltransferase, which is given as option C, is what converts putrescine to N-methylputrescine. And this acts as a precursor for tropinone. From tropinone, we get tropin, which then gets converted to litorin and hyoscyamin, and finally from hyoscyamin to scopolamine. Now, yeah, so like you mentioned, hyoscyamin 6 beta hydroxylase is the enzyme that is required here for, for a conversion of hyoscyamin to scopolamine. So structurally, if we look at it, a hydroxylase, what does a hydroxylase do? Can any of you try answering this? It's there in the name. You can maybe take a wild guess. It's called as a hydroxylase. So what do you think it would do to hyoscyamin? Okay, so a hydroxylase adds an OH group at this specific position. So that's how chemically hyoscyamin and scopolamine are different. Scopolamine has an additional hydroxyl group, which gives it additional properties. Right. The next question is identify the drug obtained through acetylation of benzyl isoquinoline alkaloid. Four different options are given here: morphine, codeine, heroin, and vanillin. This is a straightforward question. if you know what benzyl isoquinoline are. So you can take a couple of minutes and think what benzyl isoquinolines are. And from there I also have shown the structures here. Here if you see there are a couple of compounds that are structurally similar. So you need to be able to think about the structure of these compounds to be able to tell us which of are benzyl isoquinolines and which is the acetylated drug. Okay, can anyone tell us what benzyl isoquinoline alkaloids are? Among here, we have morphine, codeine, heroin, and vanillin. This is the structure of a benzyl isoquinoline alkaloid, and this belongs to morphine. Heroin is diacetylmorphine that is produced by acetylation of morphine. Here you have the hydroxyl groups that are rather acetylated both at this hydroxyl group and at this hydroxyl group. And it has been observed and proven that acetylating this enhances the lipid solubility. So these heroin molecules can pass through membranes and be more potent than morphine. Whereas the other options, which are codeine, it's an opiate, and vanillin, this is codeine. Vanillin is a flavoring compound. The next question is, name the correct precursor amino acid that gives rise to propane alkaloids. We had earlier looked at this in the form of the pathway. The four options are tryptophan, ornithine, tyrosine, and lysine. You can take a couple of minutes and then okay. Does anyone remember the pathway for propane alkaloids? Yeah, Shrista. Sorry, Shrista. It is. Yeah, Swati. Yeah, one of you can go and unmute and mention what you know about this.
Sir, I just missed one thing.
Yeah,
I became offline that you asked before the question now that morphine, some not morphine, that acetylation of benzo isoquinoline
yeah,
group. What was the answer?
yeah.
I missed it.
So
That's why I'm
this,
asking.
okay, okay, okay. So if you see here, we've given structures of morphine and heroin. And the question was which of these drugs is obtained through acetylation of benzyl isoquinoline alkaloid.
Okay.
So among these two options, morphine and heroines are benzyl isoquinoline alkaloids,
Yes.
and the only acetylated versions of these is the heroin, where the hydroxyl groups here are acetylated. You see a difference here.
So we have to remember all so many structures.
Uh,
It's really
you
difficult, no,
it,
to
it it is difficult. Yeah, I would agree, but at least maybe
activity
the
relationship.
sorry.
The structure activity relationship means we have to remember so many structures. Any easy
Uh,
way to remember?
I would say you can start by going through, you can break this into components. So here we were looking at the basic structures, like this is a phenol group. Whereas,
Okay.
what is this? Any idea? We had just mentioned it.
No, no, I missed all this. Sorry.
Okay. This was also later shown in the pathway, right? This, I think this is the troponin. Yeah. So you need to be able to break down these structures into their skeletal structures. That is what will be helpful. And if you know the skeletal structures and specific modulation, yeah, modifications like hydroxylation, glycosylation would help you tell what are the other structures. And the specific modifications can be linked to some biological function like solubility, improved bioavailability, and such things. So, if you ask me, I would say it's very difficult to remember these structures at the first go, but at least knowing the skeletal structures will be helpful. Hmm.
No, I wanted to ask,
Hmm.
given the in the questions in the offline exam, the structures will be asked. Then we have to memorize the
I
structure.
am not really sure about that part because we are involved only in the
Okay.
TA ship
The classes.
sessions. Yeah, so we are
Okay.
not really
Okay.
sure about that part. But yeah, if you know the basic skeletal structures, maybe if you're tested, it might be helpful because most of what is covered in this course is about pathways and structures. So, given the nature of the course, you might have to remember it.
Hmm.
Right.
Uh, is it yeah, propane alkaloid precursor amino acid? Ornithine?
Yeah. So, yeah. Okay. So, what is different from the what is different or specific about ornithine compared to other options given here? Any idea about that?
Uh, Tyrosine to dopamine. Uh, Lysine, propane. Yeah.
Okay,
Propane.
I've given that here. So
Yeah.
each of
Ornithine to
these,
putrescine. Yeah, to propane.
yeah. So ornithine is the one that gives you these propane alkaloids.
Yeah.
Um, Usually you won't observe ornithine as a part of protein structures. That is something specific here. Ornithine is not a part of protein structures. Proteins are made up of amino acids. Tryptophan, tyrosine, and lysine are observed in naturally occurring proteins, but ornithine is not. But regardless of that, all of these provide are precursors for other alkaloids, like tryptophan for indole alkaloids, tyrosine for benzyl isoquinoline alkaloids, and lysine for quinolidine alkaloids. Right. The next question is identify the branching point metabolite in propane alkaloid pathway where calcitogen is synthesized. You can take a couple of minutes and maybe you can start by mentioning what calcitogen is. We can take it from there. Okay. So does anyone know what calistogene is? Okay. So calistogenes are a group of non-tropane alkaloids. These also can act as glycosidase inhibitors. Now, from the name itself, you can understand that this is synthesized from propane alkaloid biosynthetic pathway. Here, propanone is the key intermediate where it acts as the branch point. At the pathway level, you can look at it here, propanone. Now, the question is to identify the branching point metabolite in the tropane alkaloid pathway where calistogene is synthesized. Like we mentioned, it is synthesized from propanone, either towards scopolamine and atropine or towards calistogenes. The pathway shown here, it produces scopolamine here, but it can also produce other molecules like calistogenes. If we look at the other options, tropine, it is derived from tropinone. It's a part of the scopolamine pathway. So once propanone gets committed towards tropine, it cannot go back to produce calistogenes. So it's the case with atropine as well. Nicotine here is an unrelated option. It is derived from nicotinic acid and ornithine. Although ornithine is the common precursor that is required for both. From this pathway, you can again see that nicotine is produced from the precursor that produces propanone. So looking at all these, we will know that propanone is the branching point metabolite from which calistogene is synthesized. Right. The next question is identify the correct pair of chemically transformed isoquinoline alkaloids among the following. So four different options are given here. Reticulin. Yeah, Swati, you can unmute. Is it about this question or the previous question? Yeah, you can unmute.
This
Is this about
this
this question?
question.
This question. Yeah, let me just finish reading and then you can share what you know. So here they are asking the correct pair of chemically transformed isoquinoline alkaloids. And they have different options. Reticulin and emetin, option 1. Emetin and oxycodone, option 2. Scopolamine, butyl bromide and oxycodone, option 3. Bu buprenorphine and oxycodone, option 4. So you can take a couple of minutes and then when you're mentioning, try mentioning what is the chemical transformation that's occurring and how the compounds are related. You can take a couple of minutes and then unmute Swati. Yeah, Swati, you can unmute, and if you know about the other options, you can mention what is the chemical transformation among these pairs. Otherwise, you can go to the answer directly. You can unmute. Yeah, you can unmute.
Uh, the answer is, I think, the buprenorphine and oxycodone.
Okay,
Both are chemically transformed isoquinoline alkaloids. Even morphine also comes under this isoquinoline alkaloid, but chemically transformed buprenorphine and oxycodone.
okay. Do you want to comment about the other options? Do you know what chemically transformed isoquinoline alkaloids are? What does that term mean?
Mm,
Are
um,
chemically transformed isoquinoline alkaloids the same as naturally occurring isoquinoline alkaloids?
no.
In what way are they different?
Chemically transformed.
You can think, okay, for example, let's look at a name here. Scopolamine butyl bromide. It has the word bromide. Do you think plants
The
can,
chemical
yeah, sorry, you can go ahead.
chemical structure information. Yeah. Please explain. Yeah.
No, no. So you see scopolamine butyl bromide and oxycodone. Do you think plants are capable of incorporating bromine into a specific natural compound? Is that possible in plants?
No.
Huh. So that is where the difference lies. So these chemically transformed compounds need not be specifically produced by plants, but their precursors will be produced by plants. So uh, here, maybe you have answered here on the basis of what isoquinoline alkaloids are, knowing bu buprenorphine and oxycodone. So what one needs to know is these are not naturally synthesized, but they are modified, right?
Yes.
So again, if we are able to know the classification of these compounds, it will be easier, like you had mentioned. Like you had mentioned, um, buprenorphine, it is again from thebaine. Oxycodone is also from thebaine, and these are chemically transformed. Scopolamine is propane alkaloid. It's not an isoquinoline alkaloid. So we go eliminate this option. Again, emitin is not an isoquinoline, uh, but it is a synthetic opioid. So we cannot consider this. And finally, reticulin, it's also benzyl isoquinoline alkaloid, but it is not directly chemically transformed. So this is also incorrect. So the answer that you had mentioned, buprenorphine and oxycodone, is right. The next question is, recently scientists have been successful to produce hyoscyamine and scopolamine from primary sugars and amino acids in yeast cell. Arrange the sentences sequentially in which the approach was made. Option 1. Enzymatic reactions in the peroxisome and the membrane of endoplasmic reticulum catalyze the conversion of putrescine to tropine. Option 2. Step 2. Glucose and the amino acid phenylalanine are converted to phenyl lactic acid glucoside (PLA) that occurs in parallel with modules 1 and 2. Glucose is converted to the molecule putrescine by metabolic steps in mitochondria and cytosolic fluid. Step 4. tropine and PLA glucoside are transported into the vacuole and together converted into litorin. And step 5, litorin is converted to scopolamine via hyoscyamine, and part these reactions occur in the ER membrane. So here if you see, the different steps of the pathway are given, and the organelle in which a particular chemical reaction happens is also mentioned. So if you recall the pathway that we were looking at earlier, based on that, you will get an idea about the order in which these compounds need to be produced. You can even backtrack it. So you can think about what produces hyoscyamine, what produces scopolamine, and maybe you can backtrack it and get an idea about the order. So maybe check the pathway once, take a couple of minutes, and think what could possibly produce hyoscyamine and scopolamine from sugars. Earlier we looked at amino acids from how from ornithine we get these, but from sugars we haven't looked at. But yeah, you can look at the pathway once and then come back and then we'll discuss. You can take a couple of minutes. Yeah, did anyone raise your hand? You can unmute if you know the answer.
Is it the fourth answer?
Okay, why would you think it's this way?
Glucose is converted. Uh, because this module number 3, that phenylalanine converted to phenylactic acid, I
Right.
think this is in module number 3. So, and glucose is converted to the molecule by vitamin A. It should be.
Okay, so
I'm not.
okay, so you said option D, right? Okay. S- Sorry, option C.
B.
Okay. So you try to trace the pathway from the last compound. We are interested in hyoscyamine and scopolamine. So hyoscyamine and scopolamine, these are produced from lithorin,
Then
right?
ornithine. Yeah.
Huh. Yeah, I mean, ornithine is the amino acid. That's at the start. But we are trying to like look from look in the reverse direction. Okay. So scopolamine and hyoscyamine are produced by lithorin. And where is lithorin produced from?
Littorin from
Phenyl lactate. And phenyl lactate is produced
lactate
from
to
phenyl pyruvate and phenylalanine,
phenylalanine.
again from glucose. So, um, by looking at the pathway, you can know with here scopolamine is produced from lithorin. That's option 5. So that's the last possible, which is why option A is right here. But if you need to look at the pathway, scopolamine is produced from lithorin. Lithorin is produced from PLA, phenyl lactate. Phenyl lactate is produced from glucose. And they say it occurs in parallel with modules 1 and 2, where glucose also produces putrescine. And here, putrescine it gets converted to propane by chemically transforming into N-methylputrescine and then propanone and propane. So what we did here was reverse or backtracking. So with that, we know that lithorin to hyoscyamine is the last step. Lithorin is produced from PLA, and this PLA is produced from phenylalanine, which itself is produced from glucose. Parallelly, glucose is also converted to produce putrescine. So this is one way to be able to backtrack and be able to understand how the pathway could possibly work. Now, other than this, any idea why this pathway will be very difficult to express in a microbe like E. coli? Okay, so does E. coli have an ER membrane? Does it have a mitochondrion? These are specific features of eukaryotic cells, and plants are eukaryotic cells, which is why it will be difficult to be able to express these proteins in E. coli and hence the pathway and the conversion. So that's why they have mentioned as yeast cell here. Right. Finally, the last question for this week's tutorial session. Identify a biologically important molecule produced as an intermediate during the biosynthesis of benzyl isoquinoline alkaloids in plants. Four options are given here: acetylcholine, gamma aminobutyric acid, glutamic acid, and dopamine. So, which of these do you think is closely related to benzyl isoquinolines? Yes, Swati.
That GABA is closely related. Am I having
Okay.
a butyric acid?
Is how about dopamine and acetylcholine? All of these are anyways similar structurally. They all have an amine group.
Yeah.
Hmm. So you would say that benzyl isoquinoline during that pathway, GABA is produced. Is that what you're trying to say?
No.
Uh,
Structurally related you asked. That's right.
okay, structurally related. That way, even dopamine would be structurally related because dopamine and GABA, in some cases, compete for similar receptors. Okay, so just a small introduction. These benzyl isoquinoline alkaloids are derived from tyrosine, and here dopamine is the precursor. Dopamine is found from L-dopa, and it combines with 4-hydroxyphenyl acetaldehyde to form the first benzyl isoquinoline intermediate. This is norladonosolin. So the incorrect options here are acetylcholine, GABA, and glutamic acid. Since we need dopamine for benzyl isoquinoline alkaloid synthesis, that is the correct intermediate. Right. So with this, we come to an end for this week's sample questions. If there are any questions that you want to discuss, we can look at that, or you can leave. I'll be here on the call for the next half an hour. Thank you.
Thank you.